Okay, so thanks very much for the invite to talk. So I'm not a CT specialist. I'm actually a physicist by training, but I work at this amazing place called the Francis Crick Institute, which is right next door to the Turing Institute. So the Turing Institute's just somewhere down here. So that's the British Library over there. And I'm going to talk about some of the image analysis. So following on nicely from Tina's talk, some of the image analysis methods that we've developed at the Crick for some of our volume data. So I thought I'd get the acknowledgement slide in first in case I'm running out of time at the end. So this is a big group effort. So the electron microscopy team at the Crick is led by Lucy Collinson. And there's a whole team of people who work in the core facility generating images and analysing images. We've also been working with the Crick's scientific computing team, so specifically the software development and machine learning team, which I appreciate we're extremely lucky to have such a thing. That's led by Amy, and Luke and Joost are the main people who worked on that. And then on the citizen science side, we've been working largely with Helen, who's now joined us as a CZI imaging fellow, and that team's led by Chris Lintop. The Crick is funded by lots of different things, so MRC, Wellcome Trust, Cancer Research UK, the 3 university partners, UCL, Imperial and King's, and obviously where possible we try and get external grants as well. So, okay, electron microscopy, for anyone who's not completely familiar with it, broadly, historically, there's the 2 sorts: scanning electron microscopy, where you look at the exterior of an object that you've coated in something metallic usually, or there's transmission electron microscopy, where you've taken a really thin slice of an object and you shine a torch beam of electrons through and you look at the shadow that's cast, and that looks at the inside of the object. But these are historically just 2D projections or slices, right? So for a long time, EM manual analysis, all of this image analysis is done manually, but the images came quite slowly, so it was kind of okay. Just the last few years, there's been a lot of developments in automating volume electron microscopy. So Tim already mentioned focused ion beam scanning EM. There's another one which we use a lot called serial block face scanning EM. So it's the same principle, but instead of a focused ion beam cutting the block, let's turn this on. So we scan the electron beam like normal scanning electron microscope, and then we have this little tiny diamond knife that comes and cleaves away the top 50 nanometers or 20 nanometers of the image, and then you just repeat, and you do that automatically overnight or over months or even years, some groups. And a typical overnight acquisition might be of the order of hundreds of gigabytes. Oops. So at the end of a run, you'll get a data cube like this, and broadly speaking, a day or 2 of doing that fills one of these. So we're sort of in the sort of terabytes per data set regime reasonably routinely now. Just a quick connection to the X-ray microscopy CT angle. So although the imaging modality is different, actually the look of the data can be quite similar. So this is a really fantastic paper by a Crick colleague, Carles Bosch, who did this work correlating across lab CT, synchrotron CT, and then volume EM. And actually you can see a lot of the data for this given sample preparation is quite similar. So a lot of the analysis techniques I'm going to talk about in EM could be applied to CT as well. Okay, so segmentation. So it's been mentioned a couple of times already. It's this process of delineating an object of interest from the background. And if you want to do true quantitative studies, you need to do that. Otherwise, you're just collecting pretty pictures, which is, very nice. But we want to quantify these things and perform large population studies over lots and lots of sample, because in biology there's so much variation. N=1 is always a risky place to be, so we want to do this as broadly as possible. Okay, and the way this is done, or has been done for a long time in electron microscopy, is like this. So somebody sits down and draws around things, and that's Still very common. Still, we do this a lot. It's the best way of doing some of these things. So this is Anne Weston, who's one of the EM team, tracing around some of the data I'm going to show you. And so this is a 2D analysis drawing over a 2D slice. But of course, the world is not 2D. So if you take a 2D view of a 3D scene, you could get the wrong impression. So you need to make sure that your analysis is truly in 3D. And so for that, we need to go back to our sample and we need to look at every single slice through the sample, and somebody traces around each slice. So this is maybe hundreds or thousands of slices to generate one object that you can then quantify. So that could be days or weeks of work to generate one thing, of which there might be 100 of them in the image you acquired overnight. So this is very low throughput. So in the past, if we wanted to do a lot of this analysis, then people just had to sit down and do it lots and lots, over and over again. Some people find it very therapeutic, some people do not. So there's a lot of interest in automating this process. And I mean, what it means scientifically is actually there's a lot of information in that data set that's being unmined because there just isn't the personnel power to go in and extract the information. So people are acquiring new shiny data sets for their latest publications, but actually there's a whole load of data, a whole load of analytical value in data that has been only treated with quite a light touch. So sounds like a job for deep learning. Everyone's turning to deep learning for these things. It's really revolutionizing the field of image analysis. And there's lots of big hopes that you can train these neural networks to generalize over different data sets and perform tasks that are essentially impossible any other way. The general paradigm is you go from the sort of classical programming method where you have some data and you try to work out what the rules are to apply to that data to get the answers that you want. What machine learning does is it turns that around. So you have the data, you supply some answers to the machine, and then it tries to tell you what the rules are that give you those answers. Which is a very nice, quick diagram there, but there's that little answers bit is doing a lot of work here. So this is somebody needs to get these answers, and if you want to do this well over lots of data, then you need a lot of these answers. And we know that humans are the best for doing this, right? So we always turn back to experts for doing this sort of analysis. And if we want to do the best possible science, then we should use our best possible analysis method. So that's humans. Okay, but we've got a lot of images, so we need a lot of humans. Okay, so this raised the question, if you want to do this sort of analysis, do you really need a PhD? Sometimes you do, identify that the endoplasmic reticulum in the sheet morphological domain involves some understanding to pick out what that thing means. But if you can phrase a question as, if you see something that looks like this, draw around it, or how many of these things are in this image, or does this image look like it's been corrupted? Very simple to explain to a person, but actually very hard to kind of systematically program a computer to be able to do. And this kind of class of human easy, computer hard problems is quite common in image analysis. And it's been approached via methods called crowdsourcing or citizen science, where you get non-experts to contribute to research by giving them questions that are approachable without all of the without the PhD and so on. So one of the early approaches to this was something called Galaxy Zoo. So this astronomers have similar problems to microscopists. They can generate millions of images automatically on these telescopes that just churn out tons and tons of data. But somebody wants to answer some scientific question, maybe what's the proportion of spiral galaxies to smooth galaxies. So the way that was done in the past was PhD students would sit down and they would go through tens of thousands of images and they would catalogue all of these images. And it worked very well, but very low throughput. So what the Galaxy Zoo team did, led by Chris Lintott, one of our collaborators there, was they had this brainwave to crop up these images, post them up on a website, and just ask people a really simple question. So the object at the center of this image, is it spiral, or is it smooth, or is it something else? And they got hundreds of thousands of people contributing to this research. And it turned out that given a good question to ask, you can get really valuable scientific input. So just a few years ago, we set up a project on the Zooniverse, which is the platform that Galaxy Zoo morphed into. And we called this project EtchACell. So what we ask people to do is we show them an image of the cell, and we give them a little tutorial, and we say, if you see this object, trace around it. So in this case, it was a nuclear envelope. So trace over the nuclear envelope in the cell and click done when you've done. We've since gone on to a few other projects. So we've now got projects for mitochondria, a VR project where we're building a whole VR representation of multiple organelles in a cell, and endoplasmic reticulum project, which has just recently started. And as well as the scientific outputs, this has been a really kind of neat thing to do as a public engagement outreach thing. So we've taken this to various events, internal exhibitions at the Crick, and events at different universities and sites around the country. And it's a really engaging way for people to see real data. So it's not just a press release of somebody in a suit saying, look how clever we are, we dreamt up this cure for something. People can actually see what scientists do day to day, and they can maybe get to trust the scientists a bit more when they can see the actual nuts and bolts of what goes on. Okay, so the main working principle is for each image, we have multiple people annotate the image. So I'll scroll through a few variants here. Some are very good, some are very bad, some are graffiti. And you can see there's a real range of sort of quality here. And there's a very important question which we had to answer very early, which was, is this good enough to do anything useful with? So, if you imagine some plot of how well an annotation agrees with the true ground truth of what's there, then our experts are presumably very high. If we have the volunteers and they're all the way down here, there's nothing we can do with that data to make it as good as using an expert. So then we really have to use experts. But fortunately, what we found was there is a good overlap. So we get some volunteers who are just as good as the best experts. We get a lot who aren't as good, but actually that's enough to bootstrap a good aggregation of the data. And one of the ways we developed for doing that aggregation was, so for example, here we can take a set of lines. You can see most of them congregate around some sort of common space. Some are all quite far away. We can look at each contour defines an inside and an outside. So we can make a heat map of essentially votes of where the contours say, I think this is inside the nucleus and I think this is outside the nucleus. If you zoom in, you get this kind of cliff edge around the outside. Halfway up that cliff, you define as the most probable position of that contour. And now we can trace that around. And I should emphasize that what we're really interested in here is the nuclear envelope, not the nucleus. So the actual membrane around the edge. So we really want the fine details of the around the outside. So we can combine the citizen scientists' outputs over several volumes. So this is a range of nuclei from that one of those large overnight acquisitions. And it's a bit noisy, but generally they've done really well. But this still isn't super high throughput. This took, many months to acquire all the data and include it all. So what we really wanted to do was feed this into these convolutional neural networks. So this is something that actually we had a really good sandwich student. So this is a industrial placement student that we had at the Crick. So he's an undergrad from Manchester, actually. And he built this neural network. So we specifically use something called the UNet architecture, which has sort of risen to the top as the preferred segmentation method, at least in biomedical images. So we scale the data appropriately, we feed it in, we train it, for a couple of days, and it produces some really nice outputs. We're predicting biological structures which are can be tricky. So here's a typical image we might want to predict the nuclear envelope for, and it's very clear in certain places, but actually in others, so like down here, the membrane is kind of oriented in the direction we've cut it, so it's very fuzzy and difficult to pick out, and that tends to trip up the predictor. So what we worked out was if we can train on one axis, one view, and then we can rotate the sample and we can predict from 3 orthogonal views and combine them. And the errors for each view are in different places. So you can scroll through. So from the X view, the Y view, and the Z view, you can see they're wrong in different places. And when you combine them, then you get a really robust 3D representation. So you can see in this image, for example, the magenta is where the red and the blue overlapped very well, but there wasn't so much green, and the cyan is where the green and the blue overlap, and so on. So you can see that this sort of kind of voting across different views was a very important step for what we're doing. So if we scroll through now one of these data stacks, you can see the expert, the citizen scientist, and the prediction. Actually, interestingly, the UNet prediction gets better than the aggregated citizen scientist data because we're able to do this tri-axis prediction, a sort of gratuitous 3D view of those combined. And one of the really powerful things about these deep learning architectures is they're convolutional, so you can apply them over kind of any size image. So this is an 8K by 8K by 500 slice stack, and it took about an hour to run through our predictor, plus a few hours of post-processing and pruning and so on. So orders of magnitude faster than it would have taken the experts to do it. Okay, so we want to know that this is working well. So we have these distance measures like the Jacquard index. So this is also called the intersection over union. So how much, so if I have my prediction and my ground truth, how much they overlap. So I want them to overlap as much as possible, but I also want as few false positives as possible. So I normalize to the total area of the two areas. That can be prone to some errors. So you get these kind of fine features. If I've got a large area that's got a very small invagination like this, and if my prediction looks like this, then its Jacquard index is very high. So that'll be a 95% Jacquard index. So that's not necessarily, if you're interested in those fine details, you want to use a different kind of metric. So maybe you want to compare the lines. So here's a line. I draw a line over it. If I get a perfect overlap, the Jacquard index is very high. But if I have a line that's just next to it, 1 pixel displaced, it now has 0 overlap. So its Jacquard index is 0, but actually maybe that's perfectly good for my analysis. So we use this thing called the Hausdorff Distance, which is confusingly the maximum minimum distance between 2 sets of points. So if I'm on the blue line, I walk along the blue line and I look at the yellow line from every point, and I mark the point at which I was furthest away from the yellow line. So that's this region at this point. This is not, this varies depending which line you start on. So if I start on the yellow line, then now the furthest point to the blue line is over here. So there's various tricks you can play to make sure that you incorporate, that you make this measure fair. So you can use undirected or average Hausdorff distances and so on. So if you're interested in specifically the contours or the boundaries of things, then this Hausdorff distance is very useful. If you're interested in the volume of an object, then something like the Jacquard index is fine if you can stand to miss a little bit of the detail around the edges. So these are now the predictions of those same nuclei, and running our metric over it, we get about a 2 pixel average distance from the ground truth. So that's a 20 nanometer or less error. And this was all written up in our publication, which went out in Traffic a couple of months ago. And explains all of the details. All of the code is linked in the GitHub repositories. The data is all publicly available. So that was the nuclear envelope. I'll quickly run through the sort of onward work that we've done with this. So we've another benefit of these neural networks is that you can repurpose a network that's quite good on a similar data set to the one you're interested in by just a small amount of retraining rather than starting from scratch. So this nuclear envelope predictor, it encodes a lot of features of EM data, whether it's nuclear envelope or noise or mitochondria. It's kind of encoded in these millions of weights inside the neural network. And you can always take that neural network and then onward train to do different organelles, for example. So we tried this with mitochondria. So we took part in the ISBE conference Mito-EM challenge for mitochondria instance segmentation. So a key word here is instance. So this is a bit different to what we did previously. I'll just explain what instance segmentation means if people aren't familiar with that. So semantic segmentation, I just say that's, that object is mitochondrion or not mitochondrion. Instance segmentation, I want to say that's mitochondrion 1, that's mitochondrion 2, that's mitochondrion 3, so I can perform more elaborate studies across the population. And so we trained our nuclear envelope neural network to perform 2 different types of predictions that gave us a good instance segmentation. So instead of having everything labeled with one label, we now have an individual label for each individual mitochondrion. Again, we can play this tri-axis prediction trick, and again, we found it very useful. So you can see there are different errors where boundaries are missed in the different views, but when you sum them all together, it makes good divisions between the individual objects. Some more gratuitous data. So this is the challenge data set. So this is a piece of rat brain tissue. So magenta is the area prediction, green is the boundary prediction of each mitochondrion. And then on the right, each the color map are the individual mitochondria as predicted by our neural network. And again, for some human brain data, and this you can see, this is really noisy, ugly data, right? There's all sorts of artifacts that the neural network is able to learn is not relevant to its prediction. So it's another thing which is a real hassle if you're trying to do this thing with traditional classical programming workflows. And we just put this out as a preprint if anyone's interested in that. Again, the data is all available freely, and there's a GitHub repo for the code. A quick note on the scaling of this. So we started with one organelle, we want to do multiple, and we've got different microscopes, we've got different sample preps, and very quickly the number of permutations explodes, and we have lots and lots of different things that we need to tune. So the scientific computing team that we work with has done some amazing work to package the code in a way that they can just automate a huge panel of hyperparameter tuning sessions to really find the best way of doing each individual thing. And it's also these network models, you can download this network model, right, and try it on your data immediately. It's all there. There's Docker containers if you don't want to install software. I think it's a great opportunity for communities and across communities, so EM to CT as well, to share these kind of models, training data and knowledge. And just to summarize, okay, so I mean, we all know manual analysis is slower than acquisition nowadays, that the microscopes are so automated that you can't possibly keep up with the manual analysis. Deep learning is currently the best in class for this. It's a very fluid field. It might change. I think everyone's waiting with bated breath for unsupervised methods to come along that maybe don't need so much ground truth. But at the moment, we do need a lot of ground truth for this training. We've shown that this non-expert etrusel citizen science approach can produce high quality annotations, which gives you enough training data to train a network that's able to generalize across different data sets. This is all cumulative, like I say, so the model can be repurposed easily. It can save you a lot of training time. It can save you having to acquire so much ground truth data. And I think a really key point, which I think is relevant as this is a Turing collaboration as well, having access to people who know how to do this in a sustainable and efficient way is critical. You know, I could go and code this up and it would kind of work, and you know, I'd probably get a paper or two out of it, but it would probably not be usable by anyone else. So having access to people who can really add the value by making it just that little bit more user friendly or that little bit more generalizable or run on different platforms, I think is really, really critical. And I'll stop there. Thank you.